All right, I'm going to get started. Good evening, everyone. Today we're going to be doing an introduction to amino acids. The way this session goes, I'm going to send the slides to everybody after the presentation, so you don't have to worry about writing down everything. The recording is actually taking a lot longer to process than I thought they were, so I may not be able to send out the recording directly tonight. If by the time I send out the slides, if the recording link's not included, send me an email and I can add the video link for you whenever it processes tomorrow. For those of you joining us for the first time, we're MCAT 101, and our mission is to provide free and fun MCAT prep materials. There's two email addresses on the slide. The first email address is our general email address, and then the second one is for me specifically. So if you have any questions, feel free to send an email to either of the email addresses. If you're not already on the newsletter or the listserv, drop your email in the chat and we'll send you weekly updates about any upcoming topic sessions and resources. I imagine the majority of the people in the chat are probably already on the listserv. And as always, if you have any questions, just drop them into the chat and I'll answer them when I find an appropriate time. All right, so today we're talking about amino acids. All right, so this is a generic amino acid. We have this part of the amino acid is called the amino group, the carboxylate group, and this part is called the amino group. So the name amino acid actually comes from the amine group and the carboxylic acid group. This carbon right here is what you learned in either general chemistry or organic chemistry as the alpha carbon. The reason this is the alpha carbon is because if you're looking at your substituents, this carbon right here is bounded to oxygen. So it's basically, it takes the highest priority. So the next carbon over is considered your alpha carbon. So your alpha carbon in an amino acid is the carbon that's bound to the carboxylic acid, and it's also bound to the amino group. And this R group here is just a side chain. Depending on the amino acid, you may or may not have a side chain. Right? And these are just basically 3D models to help you look at the amino acids. Depending on whether or not there's an R group, you may or may not have chirality involved. And we'll get a little bit more into that further in the slides. So one of the first things we need to know about amino acids is that they can form in enantiomers, which are stereoisomers who form mirror images of each other. So this is why I mentioned briefly chiral centers earlier. When we have an R group, as in the case of alanine here, our R group is this methyl group. Since the alpha carbon is bound to 4 different substituents, you can have what's known as chirality or a chiral center. That means that you can have 2 alternate versions of alanine. This is based on Fisher. Fisher came up with this naming convention actually for glyceraldehyde. It was for carbohydrates, but we also use it for amino acids. So the way you can determine whether an amino acid is an L or a D amino acid is by looking at how the substituents are ordered around the alpha carbon. So in the case where you're following from your carboxylic acid to your R group, to your amine group, if you're looking at your left palm and you curve your fingers so that you're going through in the order of corn. So carboxylic group, carboxylic acid group, the R group, and the N group. If it's your left hand that's following corn, then that's considered the L configuration. If you have to use your right hand to go through the order of corn, then you're looking at a D amino acid. So this can get a little bit tricky because there's also other ways to classify configurations. We also have another system with the R and S when we're dealing with chiral centers, but in the case of amino acids, R and S don't have a direct equivalent for D and L. So for the majority of the amino acids, L amino acids, the majority of them are S, but not all of them are S. So you don't want to memorize that L amino acids are S, because then if you look at an amino acid like cysteine, it's going to confuse you. Right. And then there's also another system that talks about bending of optical light. So those are considered plus or minus. Those also don't have, you can't tell if it's going to be plus or minus based on whether it's an L amino acid or a D amino acid. All right. So in nature, when we're talking about amino acids, we're generally talking about the building blocks for proteins. Amino acids are the monomer units. If we look here, each one of these is an amino acid, and you can combine multiple amino acids together to form a peptide or a protein. The amino acids that are used to build proteins are generally L amino acids. So people recognize that the majority of proteins and the majority of amino acids in nature are L. But there are also cases for D amino acids. They're not normally used as the building block for proteins, but they're used in specialized functions. We see them in cell wall synthesis, in cell aging, and in neural signaling. For the purposes of the MCAT, if they actually asked you about D amino acids, it would probably be just identifying that the amino acid is D based on the configuration, or it'd be a passage question. They wouldn't expect you to remember an exact amino acid and its use in the human body. So I'm just going to mention this here briefly. All right. So we can classify amino acids into three main groups to help us remember them better. All right. So I'm going to start with non-polar R groups. So again, we have right here is our alpha carbon, if I can draw correctly. Right here is our alpha carbon. So on the alpha carbon for glycine, there's two hydrogens that aren't drawn in. I'm going to draw them in here just to show you. So what our R group for glycine is just a hydrogen. So glycine is non-polar because if we look at the R side chain, it's just a hydrogen. Hydrogen is not a very polar, it's not polar. And then since we're looking at this alpha carbon right here, we have two hydrogens bound to the same carbon. That means that this alpha carbon is not bound to four different substituents. So glycine is special because it doesn't have chirality. Because in order for us to have chirality, you would need four unique substituents. All right? And glycine is also the smallest amino acid that we have. So if we start looking at a little bit larger, we get into alanine. So again, I'm going to label the alpha carbon. In the case of glycine, we had an R group that was just the hydrogen. In the case of alanine, we added a CH3 group, a methyl group. So now it's a little bit, the carbon chain is a little bit longer than glycine. It's longer by one carbon, but carbon and hydrogen, again, are also non-polar. All right. So here we added another carbon. So valine, if you're comparing to leucine, if you're comparing to alanine, I apologize. If you're comparing to alanine, you're adding an additional methyl group. So this is the alpha. If we had had just 2 carbons, one here and one here, so some people choose to remember valine based on basically this V structure. It helps them to remember that you have a V at the end. For me, I just remember that the R group is an isopropyl. So if you have propane, you have 3 carbons. An isopropyl group, you're attaching at this carbon, and you have three carbons, whichever way is helpful for you. I forgot to mention what I have listed up here. So I listed the name of the amino acids and also the 3-letter and 1-letter abbreviations. So the MCAT likes to test whether or not you know your amino acids. In order to do as well as possible on the amino acids, I recommend you memorizing the structures of the 20, some people call them essential amino acids or the most common amino acids. I recommend you memorizing the structure of all 20 amino acids in addition to their names, their 3-letter abbreviation, and their 1-letter abbreviation. All right, so now we have leucine. If we look at leucine, how it differed from what we were looking at previously is that we're adding an extra, this is the alpha carbon, we're adding an extra CH2 here. So if you remember the valine based on the V, then it's valine plus an extra carbon here. If you don't want to remember that way, you could remember that it's isopropyl with an extra CH2, or this is a structure called isobutyl, but I don't think the majority of people have isobutyl memorized. So whichever way works for you. All right, and then we have isoleucine. So here we actually have the same number of carbons and the same number of hydrogens as leucine. That's why it's called isoleucine. This is an isomer of leucine. So instead of in leucine, where we had isobutyl, so the N looked like valine, and then we had an extra carbon right here. Now we moved that methyl group so that it's here instead of here. So this is actually called sec-butyl. If you don't know sec-butyl versus isobutyl, it might just be easier to remember that it's leucine, except you don't form the V at the end. You're moving this CH2 group, the methyl, somewhere else. Right? And since so far, every one of these that we've looked at, we're only adding carbons and hydrogens, they continue to be nonpolar. Right, now we're going to get to one of our weird ones. So this is our that is not our alpha carbon. This is our alpha carbon. So our alpha carbon here is bound to carboxylate, and this is actually our alpha amine. But this alpha amine is a little bit different from all the ones we've seen before. So for amino acids, you're generally looking at having an amino group that's only bound to our carbon is only bound to the amine nitrogen once. But in this case, we're forming a ring structure, and there's actually 2 carbons bound to this amine nitrogen. So some people would actually classify proline not as an amino acid, but as an amino acid. And all the amino is really telling you is that you have a secondary amino group right here. So I'm not going to get into examples today. I'm going to mention this, but this is mainly going to be mentioned in the next session when we talk about protein structure, secondary tertiary and quaternary structure, but proline is normally located only at the end of alpha helixes and in loops because of this structure. It forms a kink when you have it as part of an amino acid chain. So the MCAT, I'm answering a question. Somebody asked if it is okay to recognize the amino acid structures instead of drawing it out. On the MCAT, you're not expected to draw anything out because it's a multiple choice exam. Okay, let me answer your first question first, and then I'll address the second question. So since the MCAT's a multiple choice exam, you only need to recognize the structures, but one of the best ways to memorize the structures is basically to draw it out. So all amino acids, you have in this case, you have an alpha carbon, and the alpha carbon is bound to the alpha carboxylate group, and amine group. So that's why we're calling it an amino acid. You always have a hydrogen, and then you have an R group. So this part of the structure, though, is the same for every single amino acid. So the only thing that changes is the R group. Drawing it out the first 1 or 2 times when you're memorizing it may be helpful. On the actual exam, the majority of the people will not draw out amino acids because you won't have time to do it. But it's good practice to draw it out so that you do memorize it, and you can figure out how the bonds are moving depending on what the question is. As for the pKa's, I'm going to show you a table later that lists the pKa's, but we're going to find that for the majority of amino acids, you just have to remember what the rough pKa value is on the carboxylate group and on the amine group. And then there's only 5 amino acids that you generally look at for the side chain pKa, and those you would need to memorize the general range for. But I'm gonna have a table later, and I'll explain a little bit more about that towards the end of this session. All right, so we're still going through the rest of our nonpolar R groups. So methionine is a little bit different from what we've seen so far, because so far we've only been adding carbons and hydrogens. So let me label the alpha amine here. I mean the alpha carbon here. We have right here is a sulfur. And this sulfur is basically, it's embedded between these 2 carbons. So it's kind of hidden between the carbons, and it's not really interacting with the outside environment in most cases. So because it's hidden in this carbon chain, we normally consider it nonpolar. There are niche cases where you could argue that this sulfur could participate in a hydrogen bond, but for the majority of the cases, you won't have to worry about that. Number one, there's no hydrogens attached to the sulfur, so it can only proceed in one direction for a hydrogen bond anyways. You're very, very unlikely to see that case, and even if you did see it, it would only be for one question. So it's probably not high yield to focus too much on niche cases of methionine. All right, now we're looking at phenylalanine. So if we look at the name here, we have phenyl and we have alanine. So this is the alpha carbon. If we had alanine, remember alanine, the R group, is basically just a methyl group. So if we ended the structure right here, this would be alanine, right? So this structure that's at the end here is called a phenyl group. So that's why it's called phenylalanine. It's alanine with this benzene ring, right? So this benzene ring is comprised again of carbons and hydrogens. That means it's also nonpolar and hydrophobic. We're gonna find that phenylalanine is one of 3 aromatic amino acids, and that provides some interesting interactions known as pi stacking that we're going to talk about a little bit in the next session. Right. Now we have tryptophan. So with tryptophan, we have again this aromaticity, aromatic structure over here, and this is considered nonpolar because though there is a lone pair on this nitrogen right here, it's actually contributing to the aromaticity here, because in order to move the electrons around the structure, this lone pair of electrons actually participating in this conjugated system. So it's not free to basically gain another charge. Right, now we're going to go into polar uncharged groups. So if we look at the alpha carbon, if we had stopped right here, this would be alanine. Now we have an OH group. So if we have an OH group attached to something, it's an alcohol. So serine is basically just alanine plus an alcohol. Or you could just remember it as adding a carbon and adding an OH, whichever way works for you. So here we have threonine. So threonine is kind of similar to serine because, again, we have this alcohol right here. But the methyl group is actually coming off. This is the alpha carbon. We have a methyl group that's also coming off the same carbon that the alcohol is coming off of. So some people may choose to remember the structure of threonine as valine with one of the CH3s replaced by an alcohol group. An alternative way to look at it is that it's serine, except there's a methyl group coming off the same carbon as the alcohol is coming off of. All right, now we have cysteine. So cysteine has what's known as a sulfohedral group. So for an alcohol, we normally have OH, but in a sulfohedral group, we have SH. So if we label the alpha carbon here, if we stopped right here, this would be alanine. And then we added a sulfohedral group. So this sulfohedral group, we'll find out, is important for what's known as disulfide bridges. And we're going to be talking about that once we talk about protein structure in the next section. All right, now we're looking at tyrosine. So here's the alpha carbon. This structure should look familiar. If we did not have this alcohol group here, this would be phenylalanine because we have the CH2 here. That up to this point right here is alanine. We add a phenyl group that becomes phenylalanine. So the difference between phenylalanine and tyrosine is this OH group in the para position. We're not covering that in this session, but para just means that it's the furthest carbon from where this phenyl ring is attached. So that's the para position, right? So if we have an OH group here, how come tyrosine is still considered a polar uncharged group? So this OH group can form hydrogen bonds. right? So that's what makes it polar, because we're definitely dealing with this alcohol here, but we have this giant ring. This giant ring is composed of carbons and hydrogens. And remember, carbon and hydrogens are hydrophobic. So since this is so much larger than the alcohol, it makes this group hydrophobic, but we still have this alcohol group that adds the polar component. All right, so what we have right here, this is our alpha carbon, right? This is carbon double bonded to oxygen. So this is a carbonyl, and we have an NH2 here. We know that the name of this structure, or we will know once we learn how to name more organic compounds. This is known as an amide. Whenever you have a carbonyl next to an NH2 like this is known as an amide. And an amide likes to stay neutral. So there's definitely partial charges that we're dealing with right here. But since it likes to stay neutral for the majority of physiological pH and what we would be considering on the MCAT, this makes it a polar group because of the partial charges, but it never forms a full charge, so it's still considered an uncharged R group. Right. So this is our alpha carbon. Instead of looking at asparagine, we're now looking at glutamine. So again, we have this part of the structure, which is known as an amide. And the only difference between glutamine and asparagine is that we added an extra CH2. It could be this one or this one, whichever one we want to think about. So glutamine is asparagine with an extra carbon link. All right. Now we're going to move to our charge groups, and there's only 5 of these that we generally look at. So earlier we looked at asparagine. Now we're looking at aspartate. So asparagine and aspartate are fairly similar. When you start reviewing the structures and drawing them out for yourself, it may help you to recognize the relationship between aspartate and asparagine, and the same relationship for glutamate and glutamine. Right? So this is our alpha carbon. This, again, is our amino group. This is our alpha carboxylic acid group. And then on the side chain for aspartate, we have another carboxylate. So aspartate is also sometimes known as aspartic acid. And a lot of textbooks don't explain the difference, so it confuses a lot of people. Right? So aspartic acid is when we're looking at the side chain that's still protonated, right? Once it donates this proton, and this becomes O minus, it's now known as aspartate. Just like when we're thinking about acetic acid. Acetic acid still has the proton. When you get rid of the proton, it becomes acetate. This is the same relationship, right? So since I drew my structures with the R group deprotonated, I named it aspartate. If I had drawn this with the H here, it would have been called aspartic acid. The MCAT doesn't always differentiate. Sometimes it does, sometimes it doesn't. So it's good to know that these two are related and why there's 2 different names for it. There actually is a difference, but that difference isn't always important to know. right? Now we're going to look at the corollary for glutamine with glutamate. So glutamate is basically the same structure as aspartate with an extra carbon. So we have a carbon here and a carbon here. So with aspartate, the alpha carbon was bounded to one carbon, and then the next carbon was the carboxylic acid carbon. So we just added another carbon link here. So this is our R group, which is a carboxylic acid group. And if we had drawn it protonated, it would be known as glutamic acid. So the side chains being protonated makes it glutamic acid. Once it donates the proton and it becomes O-, it's now glutamate. All right. So far we have two charged R groups. We have aspartate and glutamate. And those are the only two amino acids that we need to know that have that can have a negatively charged side chain. Right. Now we're going to look at more charged R groups, but we're going to be looking at side chains that can have a positive charge. So with lysine here, we have a very long carbon chain, and then we have another amino group at the very end of the long carbon chain. So at physiological pH, this is generally going to be protonated. So lysine is what some people call, I'm going to choose not to name it what other people name it. Lysine has a charged R group, and at physiological pH, it's normally protonated. Let's stick with that for now so I don't confuse everyone. All right. Now we're looking at arginine. This is our alpha carbon. So we have a carbon chain, and then we have this group at the very end with a positive charge. So this is actually known as a guanidino side chain. I do not believe that the majority of the MCAT questions would ever focus on actually knowing the name of the end of this. But when you are looking through and learning the structure, make sure that you're looking and you know that you can move this positive charge between here and here. It may help you depending on if they're asking a question about resonance. Right, so we have lysine, arginine, and then for our last one, we have histidine. Histidine is the third and last of our amino acids that have a side chain that can hold a positive charge. Right, so this is our alpha carbon. You might see this in some textbooks as a imidazole group. All right. All right, so this is glycine again. So glycine, remember, had hydrogen as its R group. The reason I chose to show glycine here is because I wanted to talk about Zwitter ions. So amino acids, frequently when you're learning about amino acids, people talk about Zwitter ions, or sometimes they might mention that amino acids can be dipolar, right? That's just referring to the fact that you have a carboxylic acid group that can be deprotonated, and then you have an amino group that can be protonated, right? So you have both a positive charge and negative charge on the same molecule. So overall, this molecule is neutral, but there is a positive ion and a negative ion on the same structure, and that's what's known as Zwitter ion. So this is basic vocabulary and actually comes up quite often, even on the MCAT. All right. Somebody asked earlier about pK values. So I've included a pK chart out to 2 decimal places. And in most cases, I would not bother memorizing all the slight changes in pK. So right here, we have the pKa for the alpha carboxylate group. So if we're looking at an amino acid, we have COO minus, that's our carboxylate group, and this is bound to our alpha carbon, right? So if we look at the pKa for this group right here, and we look straight down for our main 20 amino acids, we'll notice that this pKa value is around 2 for all of them. So that's honestly all you need to know. It won't help you get any more questions correct if you remember that glycine is 2.3 and proline is 1.99 or histine's 1.8. For the purposes of the MCAT, it's enough to know the ballpark figure. And then our alpha carbon is also bound to the alpha amino group. So for the purpose of the MCAT, if we look straight down, we see that the value fluctuates a bit. It would not hurt us at all just to remember 10. So the pKa's are important, but if they're not going to ask you to whether the pK of the alpha carboxyl group, the slight changes from one amino acid to the next. I'm not going to say nobody. There are some people who actually have these memorized for the different amino acids, but you're not expected to have these memorized. Now, the pKa's for the side chain, I would recommend memorizing these. So the 5 amino acids that I have at the bottom are the 5 that I went through that you would most likely see on the MCAT with either having a side chain that's positive or negatively charged. So you have aspartate and glutamate, which both have carboxylic acid side chains. That's why their pKa's are in the 4 range. So for these two, you'd be fine just remembering that if you have a carboxylic side chain, that the pK is around 4, right? And then for lysine, arginine, and histidine, you just have to remember these. Whether you choose to remember lysine as 10 or 11, it wouldn't make a difference. I have it memorized as around 10.5. I haven't checked this value in years, but if you use 11 or use 10, you wouldn't get anything wrong. For arginine, you can remember 12. For histidine, some people say 6, some say 6.5, some say 7. Histidine is a fun one because histidine, if you think about physiological pH, people sometimes say 7 or between 6 and 8. Histidine is so close to physiological pH that it's the amino acid that you're most likely to see with its side chain protonated and deprotonated, right? So key highlights from this page, just to reemphasize. pK of the alpha carboxyl group for all the amino acids, memorize it as 2. pK for the alpha amino group for all amino acids, memorize this as 10. And then you have to memorize these 5 pKa's for the side chains of the amino acids that can hold a positive or a negative charge on their side chain. So for aspartate and glutamate, or if we're looking at their protonated forms, aspartic acid and glutamic acid, we can remember their side chain pK as being around 4. And then you just have to memorize lysine, arginine, histidine pKa's. So if you memorize all of these. That's only 1, 2, 3, 4, 5, 6. 6 pKs you have to remember for amino acids. It's not the most difficult thing to memorize. When you're working through your practice problems and preparing for your MCAT, you're going to see just how useful it is to be able to pull the structure of the side chain that you've already memorized and to know the rough pKa range of the side chains. All right. So now that we looked at pK a little bit, let's look into actual ionization. So we're going to start with glycine again, because glycine is easy. Glycine doesn't have a charge on its side chain, so we only have to worry about 2 pK starting out. So if we're looking at low pH, so let me label this. If we're going from low pH to high pH, let's see what's happening here. So at very, very low pH, all of our groups that can be protonated are protonated. Because what does low pH mean? Low pH means that you have a high hydrogen ion concentration, right? Because pH is the negative log. So if you have a high value of something and you take the negative log of that value, it's going to give you a low number. And then at high pH, you're going to have lower H+. So when you have very high H+, you're going to have a protonated amino group. You're going to have a protonated carboxylic. Well, this would be carboxylic acid, since I still have the H on it. So your carboxylic group will be protonated and your amino group will be protonated. Since this is neutral and this is positive, we have a +1 charge here, right? So if we increase the pH though, that means we're lowering the hydrogen ion concentration. The next group that's going to change is going to be our carboxylic group. So we deprotonated our carboxylic acid, and when our carboxylic acid group loses the H+, it becomes carboxylate, right? So now we have a negative charge and a positive charge. This is the Zwitter ion, right? I should have abbreviated so I wouldn't have to write this whole out. So this is our Zwitter ion. This is dipolar. We have 2 poles with opposite charges, but the overall charge of this molecule is neutral. Right, so let's call this 0. If we increase the pH even more, then we're again decreasing the concentration of H+. Once we get to a certain value, our extra H+ from the amino group comes off, and we're left with a neutral charge. We're left with NH2 as opposed to NH3 plus attached to this carbon. So this becomes a -1 overall charge. So as we're increasing pH and we're looking at an amino acid, the overall net charge decreases, right? So we start with our highest positive charge possible. For glycine, since we don't have any other groups on the side chain that could be protonated or deprotonated, we're only looking at the amino group and the carboxyl group. So you start with everything protonated, and then you deprotonate each group based on their pKa's as you increase pH. So I'll put this here as a reminder. So what's the Henderson-Hasselbalch equation? And what does this equation have to do with ionization? Right. So when we're learning about acids and bases, we learn this equation called the Henderson-Hasselbalch equation. It tells us that when pH is higher than pKa, we have more of the deprotonated form than the protonated form of whichever species we're looking at, right? So this pKa right here is the pK for the carboxylic acid or carboxylate group. We generally remember this as being around 2. So when our pH is equal to our pK, so if our pH right now was around if our pH and our pK are equivalent to each other, what happens? If we look at the Henderson-Hasselbalch equation, if these are both equal to each other, then pH minus pK would be 0, right? So we would have 0 is equal to log of our deprotonated form over our protonated form, right? And since I'm trying to get rid of this log, I can do 10 to the 0 and 10 to the log. This is anti-log, that's what I'm doing right here. So 10 to the 0 is 1. Log is cancels with the 10. It's going to be a minus divided by HA. Or if we multiply both sides by HA, we find out that when pH is equivalent to pKa, we have the same amount of the protonated species as the deprotonated species. We also could have known this just based on the definition. When we learned definitions of pKa, the pKa was described as the pH where we have the same amount of our protonated and deprotonated species. But you can look at the equation and figure that out also. So when we're looking at the pK of the carboxylate group, if our pH is equivalent to this pK, so if our pH right now was equivalent to 2.34, then half of our glycine would be in this form, and half of our glycine would be in this form, right? So this has an overall +1 charge. This has an overall charge of 0. Plus or minus 0 are both 0, so it really doesn't matter how you label that. So if we're at this pH, then we would have equal amounts of the form of glycine with a +1 net charge, and we would have the same amount of +0 overall charge, the Zwitter ion form. So if we were asked on an exam, for example, at this pH, what is the ratio between these two? We know that it's 1 to 1. Other ways they could ask the question is, what is the average net charge at this pH? So if half of mine is +1, and then the other half of my molecules is +0, we average those, and the average net charge would just be +1/2. That's another way they could ask the same thing, just to test, basically, if you understand the Henderson-Hasselbalch equation, or if you understand pK and pH relationships. Right? Now let's look at this second pKa. Right? So this pKa is 9.6. This pKa is actually for the amino group. We could have memorized this as 10. For glycine, from the table that I provided previously, it was labeled as 9.6. So at pH is equal to 9.6. Since 9.6 is the pK of my amino group, I would have half of the glycine in this form with a net charge of -1, and half of the glycine in the Zwitter ion form with a net charge of 0. All right, so this brings us to a very useful point. I'm not talking about electrophoresis today. I will talk about it in the next session, but I'm going to give a brief intro on isoelectric point. So if we're looking at this molecule of glycine, at the isoelectric point, the isoelectric point is basically defined as the pH where the molecule carries no net charge, right? So pH where no net charge, and we're going to pretend that everybody can read my handwriting. So why would this be useful? Well, if we're looking at electrophoresis, we could use pH to separate different amino acids or different proteins based on what's known as the isoelectric point. So, for instance, if my isoelectric point for glycine is this for glycine? Yes, it is. So I calculated the isoelectric point here. But since the pi for glycine, the isoelectric point is 6, if I increase my pH above 6, then glycine in a gel electrophoresis system would continue to move towards the anode or the positive electrode until the pH is equivalent to the pi. Right? If pH is less than the isoelectric point, then the molecule would continue to move towards the cathode or the negative electrode until pH is equal to pi. So what actually happens is if we have multiple molecules, all with different PIs, we can use pH and we can look at how far on a gel these migrate to figure out what the PI or what the identity of the molecules that we're looking at. And we're going to be looking at a little bit at these examples in the next session. For now, what I really want you to know is how to calculate the isoelectric point. So the isoelectric point for glycine is actually really easy because we only have 2 pKa's. So if we're looking at the point where there's no net charge, then we're really just looking for what pH do I have predominantly the Zwitter ion form. This is simply just the average of these 2 pKa's. It's going to be just in the center. So for an amino acid who doesn't have an R group that can be charged, we just look at the average of the 2 pKa's. So since all amino acids have the same carboxylate and amino group, and they only fluctuate slightly, for any amino acid that we're not concerned about a charge on their R group, the isoelectric point is going to be around 6. And we'll see that when we take a look at our table again. All right. So now let's think a little bit about histidine. So for glycine, we only had 2 pKa's. For histidine, these are the 3 pKa's for histidine. So this is the R group pK. That's the imidazole ring right here. This is the pK that we have memorized as 10, the amino group. And then this we have memorized as 2. NH3 plus. So we have 3 pKa's. So in this case, how would we figure out at what pH this molecule of histidine would be neutral? Since we have more than 2 pKa's, it requires a little bit more thinking, right? So if we had unlimited amount of time and we were only concerned with getting the correct answer, I would just write everything out. So how about we rearrange these pKa's in ascending order? So we have 2, 6, and this has 9, but since I'm telling you to remember 10, so we don't have to remember what this value is for every single amino acid, we can just think of it as generally being around 10. If we look and have 3 pKa's, we can start comparing at what pH we're going to have which groups protonated, which groups deprotonated, right? So if we had every single group protonated, so if this was protonated, if this was OH, there's no charge. If this is protonated, that's NH3 plus. This is protonated, so that's +1 charge, +1 charge, -1 charge. So at very low pH, we have a +2 charge, right? Because this gives us +1, this gives us +1, and since this doesn't have a charge on it, this is just 0, right? So if we increase our pH, when we hit 2, what happens at pH 2? All right, so this is coming back to the Henderson-Hasselbalch again. 2, remember, is our pKa. So if this is the pKa, at this pH, we would have equal amounts of 2 different forms of whatever we're looking at. Since this pKa belongs to the carboxylate group, at pH 2, we would have half of our histidine with this as OH and half as this is O minus, right? Since we're increasing pH though, right here we would have more of the protonated form. Once we pass pH 2, we have more of the protonated form. Right? So if this group is protonated, then it would not have a charge. But if we're increasing the pH and this becomes deprotonated, then this has, can I change the color of this? Right? So if my OH group becomes O minus, I now have a -1 charge. I still have a +1 charge here, still have a +1 charge here. So at pH 3 or 4, I'm getting more and more of the form that has a +1 charge, right? Now, if I continue to increase my pH, when I hit pH 6, this is again another pK. This pK belongs to the R group. So at pH 6, I'm going to have half of my histidine with this group protonated and half unprotonated, right? Once I go above pH 6, I'm going to have more of the deprotonated form. So if this becomes deprotonated, then this would have a charge of 0, right? This still has a charge of +1. This still has a charge of 0. So overall, I feel like I wrote something down wrong. I'm going to change colors because I'm, all right. So if this becomes deprotonated, we have +0 here. This is +1. This has become, this pK right here was the carboxyl for the carboxylic group, right? So it's already been deprotonated. So this is -1, right? So we have +0, +1, -1. So here, our overall charge is +0. And then this pK, I'm going to change colors again. pK of 10 is for our alpha amino group. So once we go above pH 10, we're going to have more of the deprotonated form. So we're deprotonating this group. This becomes +0, right? And we still have the negative charge on the carboxylic acid group. This has already been deprotonated, so this is still 0 for an overall charge of -1, right? So let's think back. What were we trying to solve in the first place before I went through all of this? We were trying to figure out how to find the isoelectric Have a good night, everyone who's hanging off. We were trying to find the isoelectric point for histidine. So at the isoelectric point, that's the pH where we have an overall net charge of 0, right? So that's going to be, since we drew all this out, we know that's going to be between 6 and 10. Since we know it's between 6 and 10, we just average these two pKa values. And if we average these two pKa values, that would give us 8 as the PI for histidine. So the actual PI for histidine, if you look in most textbooks, it's going to be around 7.5, but that's because we were using rounded numbers. If you get 8 instead of 7.5, that's not going to change your answer on the MCAT. So I would not recommend drawing out the structure of the molecule to solve this on the MCAT, but you can reason using the pKa's and the groups. And once you've gone through this exercise with 2 or 3 different amino acids, you'll have it down and you won't have to draw out this whole entire thing to think about it. Normally, I just write out the pKa's, and from just looking at the pKa's, I can figure it out. Another way you could do it is to figure out which of the 2 pKa's are closer to each other. But if you did that, this would confuse you since the distance between 2 and 6 and the distance between 6 and 10 are the same. Will we go over practice questions on this topic in later sessions? Yes. So I'm trying to stay as close to an hour as possible. So this session is mainly an intro to amino acids. On the next session, I'm going to be doing protein structure in general, and then towards the second half of the second session, there will be practice problems. I'll try and throw another one like this so we can go over it. And then if you have any other questions, you could also send me an email. How do we calculate like this on the MCAT? It's going to take me a while to calculate this. So on the MCAT, another thing you could do, let's go to the next slide here. Let me change my pen back. All right. So for anything that doesn't have a pK in the side chain, the PIs are going to be around 2 plus 10 divided by 2, right? So PI for most amino acid is going to end up being around 12 divided by 2 on 6. The only ones that we really need to have down are these 5 down here. If we look at the pKa of the side chains, aspartate and glutamate are both around 4. Right? So what you would find out after making a chart for either one of these, if I listed the pKa's, so this is for my carboxylic acid group. This is for my R group, and this is for my amino group. You'll end up finding out after solving this that the PI for both glutamic acid and aspartic acid is around 3. Right? So if you had gone through and basically created this chart once or twice, you'll notice a pattern that for the 2 amino acids with a side chain that can be negatively charged, the pi ends up being around 3. So one way to do it would just be to memorize it, or the way I have it memorized is I'm looking at the differences between the 3 pKa's, right? These two groups are, these two pKa's for these two groups are closer together. So I know that the neutral point is between these two. So I just take the average of 2 and 4, right? For the positive ones, for the amino acids that can be have a side chain that can be positively charged, the PIs are going to end up being between the upper region. So let's see, which one do I want to pick? Let's do arginine. So for arginine, right, we have 2, 9. We're going to do 10 just for consistency. I'm listing out the pKa's for arginine. So if we memorize the pK of the side chain to be 12, again, I can use the same trick here. These two pKa's are closer to each other, so the neutral point is going to be between these two. So pK, the pi is around 11. I believe the actual value is close to around 10.8. So for our purposes, 11 is enough. So you could just remember that for the ones with the negatively charged side chains, PIs are in the region of 3. For the ones that are positive, it's going to be between 10, because that's the substrate, not the substrate, the alpha amine, and whatever the pKa of the side chain is. So for histidine, we have 9 and 6. 9 and 6 would be 15. 15 divided by 2 is 7.5, and that's actually right around where the pi for histidine is. For lysine, we have 10.5 for the side chain. And here it says that the alpha means 9, but if we remember 10, it would still be enough. So between 10 and 10.5, that gives us that the pKa for the side chain of lysine is around 10. The pi is actually 9.5, but 10 would have gotten us close enough. All right. So we're done with the content for this session. Does anybody have any questions that they want me to go over real quick based on what we've covered today? Please confirm that you said to focus on the pKs that are closest in value. Oh, sure. Now I wish I had just made a slide for calculating pKs for the, all right, so if we're looking at acidic side chains, I don't want to use that word. If we're looking at side chains that can hold a negative charge, so negative, let's do negative R group. So that gives us aspartate and glutamate. The pi values are, the neutral point is between a pK of the alpha carboxylic group and the R group. And if we had memorized that the pK of the side chain was around 4, right? The PIs for both glutamate and aspartate end up being around 3. Right? Now, if we're looking at a positive side chain, I'm going to change colors. If we're looking at a positive side chain, right, that's lysine, arginine, and histidine, then the PIs are going to be between the 2 higher pKs. So we have the lowest pK is 2 for all the carboxylic groups. For all the amine groups, we're looking at around 10. And then the pK of the side chain, right? So since we're looking at the positive ones, we don't have to worry about this pK. We just need to look at the pK of the amino group and the side chain. So whatever the side chain is, for histidine, we would have 6. So for histidine, 6 and 10, if we took the average of that, the PI is around 8. Whereas for arginine, if we use 12 for the side chain, our PI is around 11. So, I mean, it's the 5 that you'll see most frequently everywhere because in nature, you only use primarily 20 amino acids. I mean, when you're learning about metabolism, you find out that there's other amino acids like ornithine and other variations, but they're all built off of these 20. The last 5 are the 5 that are normally considered to have a charged side chain. You also have tyrosine and cysteine, but you don't see these as much. And if you did see it on the MCAT, it would be very explicit how they explain it. It would probably be explained in the passage, or they would draw the mechanism for you. Yeah. So as you guys get a chance, remember to go through, memorize all the amino acids and their abbreviations, both the 3-letter and the single letter. And this will help you very much on all the sections of the MCAT that could have amino acids. Memorize which ones can have a charged side chain. There's only 5 of them, right? Memorize which ones are considered nonpolar, which ones are considered polar. We'll review in the next session. I will talk about all the fun exceptions when we're dealing with amino acids. For now, tyrosine, tryptophan, and phenylalanine are aromatic. So different textbooks. I'm answering a question that's been here. Somebody's asking, do you recommend any resource for memorizing? I find different textbooks draw them differently, which has been confusing. Find whichever one works for you. The reason it's confusing is because people don't consider the pH when they're drawing amino acids. Here, let's look right here. I'm gonna clear this slide. So at physiological pH, this is the structure of glycine, right? So, you know, we have our pK right here is around 2, and our pK right here is around 10. Physiological pH, some people might call 7.4, some people might say it's between 6 and 8. Between pH 6 and 8, our carboxylate group is deprotonated and our amine group is protonated. For some reason, textbooks, whenever they introduce amino acids, they choose to list this form, but this is not the form that we normally work with. I mean, you need to know which form it's going to be in at every single pH because you get a lot of questions about that. But when you're in this range, you're not going to have this group protonated for the majority of pHs. The only time this group is protonated is when you're below pH 2. And then let me mention again about glutamate and glutamine. glutamate and aspartate. When they're named like this, when it's ending in 8, that means it's already deprotonated, right? So if you were still protonated, it would actually be called aspartic acid and glutamic acid. And some books don't differentiate between the naming conventions, but this is an acid because it still has the proton that it can donate. Once it's donated the proton already, it's not an acid anymore, right? So this is the 8. All right. We've switched over from using GoFundMe to PayPal. So if you'd like to help us run these sessions, please consider donating to PayPal. As always, this is not mandatory. All right. So thank you for attending, everybody. I'm going to send out these slides to you probably within the next hour or so. Will the PayPal link be pasted on the Facebook group? I'm not the person that manages the Facebook group. We do plan on changing out that link, but I don't know when it's going to be changed. The PayPal group will be in the email, and it'll also be on the slides that I'm sending out, though. Next Friday, I'm holding another session on amino acids. It's not going to be the same material. I may review briefly if we have the time for it, but then I'm going to be looking into amino acids in terms of how they form proteins through, and then we're going to be looking at a little bit about hydrolysis and dehydration, as it has to do with forming the peptide bonds. And then we'll work through some sample problems. I think that one of the other tutors also wanted to co-lead the session. We'll see once it's finalized. So we will have a session. I'm not sure if it's going to be a co-led session yet or not. All right. I'm going to stop the recording, but I'm going to stick around. So if you have any questions, I'm still here to answer them.